Well, welcome to Eastern Carolina for kind of an unusual day. For one is, it's in the middle of the afternoon. It's a comfortable temperature in the middle of July, and it's a cloudy day and it's not about to rain. It usually rains cats and dogs, and then it clears up and you have humid weather. So that's Eastern Carolina. Here I am sitting between some of my friends of Mexican tarragon, elderberry, and we're working on our garden. Got a forest of jalapenos coming in, and behind that a forest of basil in a couple of various bins. So things are adding up. What I wanted to talk to you about today was kind of the miracle that's happened in the last 15 years that kind of just gets forgotten. You know, between the years of 2003 and 2006, two amazing studies came out that changed everything in terms of nutrition and hopefully in medicine. Medicine's driven by a different political party, but the idea that these studies came out, and one is I want to tell you about a little bit, and I've talked about a lot before. They're both out of North Carolina. One was out of Duke at the time in 2003 by Dr. Randy Judi, and the other was 2006 by Dr. Zeisel out of the University of North Carolina. They both are currently at the University of North Carolina, and we're in North Carolina. So what Dr. Judi's was, which was so profound, is he took this thing called these Agouti mouse, and these mice that are bred to be yellow and obese and basically prone to cancer. That's all you need to know. And generation after generation, that's what you'll get. And so he thought, well, why don't I just give, these mice, before they get pregnant, why don't I give them methyl donors, otherwise known as B12, folate, choline, and see what happens. And so he did, and he found out that the generation after that were not sick, fat, yellow mice. They were healthy-looking mice, which is kind of a reddish-brown, lean, and so on, and they lived healthy lives. So that was so impressive because it was so easy to see the results. And it was done again and again and again and again and again. And then 8 years later, he did another variation of that. And what he did, he had the same yellow, fat, prone to cancer mice, and he gave them a toxin throughout their gestation. And the toxin was what you get in plastics, BPA, bisphenol A. And so he had two groups of mice, and one group, both groups he gave that toxin to. And one group, he gave all these methyl donors. We'll call them supplements, vitamins, right? So that's the B12, folate, choline, SAMe, and that's pretty much it. And they turned out normal. So he goes, well, Shazam, look what methyl factors do. So the whole world became methylation crazy. They go, wow, we need to have this stuff. This is really cool. It's going to make everybody healthy. Well, it doesn't quite work that way. But what did happen is that we all started thinking about how you can turn genes on and off. The whole methylation, which had been known before, became kind of more common knowledge, almost a common way of discussion, not just among scientists, but a way of looking at health. All right, 3 years later, 2006, again at University of North Carolina, we moved from mice to humans. And so this is a brilliant study, and it's going to humans, which is always a little dicey. So roughly it's about 60 humans, 57, and it was about a third men and about a third postmenopausal women and about a third premenopausal women. And what they did, they focused on one thing, choline only. And so they started them all off at what the NIH had established at what they call an adequate dose. 550 milligrams per 70 kilograms. So about me, which is 155 pounds, my size. And so they were given adequate amount of choline just to start this experiment, all of them. And then they were put in depletion. They dropped it down to far lower amounts of choline to see what would happen. They would follow their liver enzymes and their kidney enzymes and so on. And what they found was this, that the men had the hardest time. More men suffered even at the higher level of choline. They weren't getting enough choline. So this was on a per body weight basis, right? I don't know if it was ideal body weight or just regular body weight. So they suffered the most. How do they know? Their liver enzymes, it was mostly their muscles. Muscles, you measure that through kidney enzymes. So, well, that was interesting. What about the pre and postmenopausal women? The premenopausal women didn't really suffer at all. They had moderate and it's all statistically looked at, and they're followed with all their blood work and so on, they had an okay time. And so it wasn't until all that choline was dropped down to a depleted level. They stayed at this depleted level for a number of months, and that they would follow the blood work, that the premenopausal women did okay, but some started to show some problems. Whereas the postmenopausal women were somewhat like the men, were having problems. What does that mean? That means that most women, premenopausally, in essence, can make their own choline. They're not dependent on their diet. That was a huge breakthrough. So we went from mice to humans, focusing on choline. So human studies are really hard to do, by the way. You get all these permissions and so on and so forth. So to actually get people to deficiency state and and then after they get to deficiency states, once they're there, you have to bring them back, obviously, to a healthy state, to replete them. And it took a lot longer for men to get repleted because they needed higher levels. So what that means is, the takeaway there is, even those established levels of what they call adequate intake, or AI, are not high enough for most men. And that for most, pretty much all menopausal women, need to increase their choline. So it has to do with requirement. All right, well, we got to talk about choline for a second, because choline has like 3 functions. And we're not going to go deep into this, but I've mentioned this in some of the videos, and it is important to know. And I think you should know it. So one is that choline goes through a number of biochemical reactions that are controlled by enzymes, and these enzymes have genes and mutations. So for some people they don't work at all, but for most people they work okay, right? And so choline makes for phosphatidylcholine in your body, which is about 95% of all your choline in your body is in lecithin, phosphatidylcholine. It also goes into methylation, gets oxidized into methylation, and it's a big helper. And that's really important to turn genes on and off. But the third function that you don't hear much about is that it actually is a precursor. It goes in to make an essential fatty acid, an omega-3 called DHA. So you probably know fish oils, which is EPA, and DHA. It makes DHA. All right, so why would you care about that? All right, big picture now. We think of choline deficiency as, what problems would it cause? It would probably cause a acetylcholine deficiency. All right, what does that mean? Acetylcholine is the dominant neurotransmitter in the brain. That would probably have to do with brain problems like dementia and Alzheimer's and et cetera, et cetera. It would also have to do with muscle. We saw that. So not enough choline would be muscle problems, because acetylcholine is the dominant neurotransmitter for muscle contraction. And then it's very much involved in the liver. So if you're deficient, you're going to have liver problems, muscle problems, and basically brain problems. All right, so what do we mean by brain problems and this DHEA thing that I'm talking about? Well, one of the things that's sort of obvious with dementia is what they call gray matter volume. The size of the brain gets smaller, shrinks, disappears. And of course, you've seen some of these pictures, which are autopsies, that shows that a lot of the brain has disappeared. So this gray matter volume gets smaller and smaller and smaller. And it's associated with dementia, and certainly associated with, even more so, with Alzheimer's, which is part of dementia, of course. So that's another reference to, gosh, choline does these 3 things that, it helps the DHEA, which lack of is associated with gray matter volume decrease, hence dementia, and as well as acetylcholine for brain and muscle. And so it's like, that's amazing. So now we're really specific to this one methyl donor, if you will, this one nutrient that does so many different things that just became an official sort of nutrient in 1998. So it's very important. I just wanted to give you a visualization of the journal research of 2003 and then in 2008, and because it really set the stage for people to think differently. All the little pieces of the research were available, a mutation here, a mutation there, they knew about, but it didn't come together as profoundly as it did in this particular simple, elegant, as they would say, experiment that has been done probably thousands of times since then. It was just so visual inaccurate. All right, so here you go. Here's the agouti mouse. It's fat, it's obese, it's prone to various cancers. That's what a normal mouse is looking like. And you can come back and look up the various articles should you want to. Okay, so from large, obese, diabetic, yellow mouse prone to cancer, the agouti mouse. You buy them. They're like bred to be that way. They're an experimental mouse. That's how mice are, by the way, in the labs. Okay, so they gave them both mouse chow, except for one. He gave them B12, folate, SAMe, which is related to choline more than anything else, but it was a very famous. I'll show you what these are in a second. Choline and trimethylglycine, which is also known as beta-ine, and that is a metabolite of choline. So it's kind of choline-oriented, folate, and B12, okay? Just want to give you a peek at this. So this is what we're talking about when we talk about methylation. It's a core. You really need to start bringing in some of this information and maybe not know every little spot here. This comes from SpectraCell. I use their diagram because I like its elegance, its simplicity. And this is about methionine cycle, otherwise known as methylation cycle, folate acid cycle, and this is about neurotransmitters. I'm not going to go that far, but I wanted to point out these are the methyl donors. That's choline. This is B12. That's folate, otherwise known as B9. And these are the three that he had given and gave SAMe. There you go. So those are the ones that he had given to the mice. So these methyl donors are not that fancy. You can get them at any good pharmacy. You can get them from your doctor. So there's beta-ine, that's right. That's trimethylglycine. That's a metabolite of choline. That's SAMe. And here's B12 and folate. That's all he gave. Put it into the mouse chow. Boom. All right. Now what happened? They all came out healthy. All the mice were healthy. They were variations, but they were basically all healthy. So in one generation, Shazam, feeding the mother methyl donors, you might want to say feeding the mother a nutritious diet with methyl donors, suddenly turned on the appropriate genes and turned off the other genes. That's what methylation's about. So there you go. And then he did another study 8 years later in which he did the same setup, same agouti mouse, right? And to this, he did the same thing. They both had the same chow, but he gave them both a toxin, BPA, bisphenol A. That's the one you get from plastic bottles. And what was the outcome? Well, he expected all these things to get plugged up, but he did the same thing, same methyl donors, same situation, same healthy mice. So this means it even helped with environmental toxins. And this was so profound. The second, 2008, study was so profound, it really opened up, and he coined it environmental epigenetics. So how environmental toxins go in and turn off some genes, turn off other genes, which was the situation here. But when you're given the appropriate nutritional supplements, this is just, we don't know about any of the mutations or anything of that. It's just you gave them all the methyl donors, those, the big three. Technically there's big four, but big three. And this is the results you got. That's phenomenal. So I wanted you to sort of see that picture, to know this is what's happened. This totally changed the stage in terms of how we look at nutrition now. Clearly, folate, B12, choline, SAMe, and the derivatives thereof, like beta-ine, all rose to the top as being very important. The problem with that, when we talk about this, you have an oversimplistic understanding, many people, and they go out and buy just those supplements, and they get themselves into trouble because the world did become methylation crazy after that, and it didn't work for everybody because you needed a larger, on a foundation of whole foods, egg yolks and liver. So here's the other study that I've told you about with Dr. Stephen Zeisel from the University of North Carolina. And this came out in 2007. It was actually put together in 2006. So really about the same era. Within 3 years, 2 phenomenal studies came out, briefly popping through this, just so you get a visualization. So here's the background. Although humans require dietary choline for methyl donation, membrane function, second function, and neurotransmission, meaning acetylcholine, choline can also be derived from de novo synthesis. In other words, you can make it up yourself. Those women with high estrogen that are working fine and don't have these mutations. The synthesis of phosphatidylcholine, which is upregulated by estrogen. A recommended adequate intake, AI, exists for choline. However, an estimated average requirement, that's another way of looking at it, has not been set because of lack of sufficient human data. So the objective of this particular study is to evaluate dietary requirements for choline in healthy men and women, and to investigate the clinical fallout, sequelae of choline deficiency. So there's still a lot of unknowns relative to choline. It's not wrapped up in, hey, we need to take it. What form? How much? When? It's still unknown. Okay, so the design is this. 57 adults, call them 60. 26 men, 16 pre-menopausal women, 15 post-menopausal women, kind of 50-50 there, with fed a diet containing 550 milligrams of choline. So that was set by NIH by saying, hey, that's the upper bar for what we believe mostly men need. All right, so they're given that, and that's for a 70 kilogram person, so it's 155 pounds, right? So that's your average male size. They put it on a per body weight basis. So they did that for 10 days, followed by dropping it down to 50 milligrams, far less, with or without folic acid supplementation for up to 42 days. I'll show you what this looks like in a second. So the reason they did with and without folic acid, because they wanted to be able to remove the effects that folic acid may have or not have, because that was one of the methylating factors, right? Methyl donors. Okay, subjects who developed organ dysfunction during this diet had normal function restored after incremental amounts of choline were added back to the diet. So in other words, they're following labs, they're looking at liver enzymes and everything else, and so, we found liver dysfunction, whether it's muscle or liver. Then they gradually brought them up to the level of normal. So they didn't leave them hurting. And blood and urine were monitored for signs of toxicity and metabolite concentrations, and liver fat, because they're looking for fatty liver as well, was used by MRI. Okay? All right, so here's what that looked like. So for 10 days, they're all in 550, pretty much a lot. For most people, it's probably a lot. And then they were depleted for an average of a month and a half. And then they were brought up to normal again. And so I'll just leave it at that. That was interesting. So now you get a visualization of what it is. But here's the results. When deprived of dietary choline, 77% of men, 80% of postmenopausal women, call that the same amount, developed fatty liver or muscle damage, whereas only half that amount, 44% of pre-menopausal women, developed such signs as organ dysfunction. Moreover, 6 men developed these signs while consuming 550 milligrams of choline. So even though they were having their top amount of choline given in their diet, it still wasn't enough for them. They were still having muscle dysfunction in these particular cases. And the folic acid supplementation did not change anything. So they had to be brought up even higher. That's why it's still up in the air. How much choline do we need? Okay, so the conclusion is subject characteristics. Example, menopausal status modulated the dietary requirement for choline, and a daily intake of the current adequate intake was not sufficient to prevent organ dysfunction in 19, that's roughly out of 57, that's over a third, that's about a third of all the people it wasn't adequate for. And this was basically showing about the brain dementia, how what you have is brain dementia. This is a number of gray matter atrophy, so the more and more they get in dementia, more the lacking of gray matter that they have. It's worse in Alzheimer's, and it really isn't a factor in vascular dementia. I wanted you to see that so you had a visualization of that. So once you get into human research, that's really pretty restricted. You need a lot of permissions, and so if you're going to take a population of people, nearly 60 people, down to deficiency levels and hold them there for a while and see what falls apart, what dysfunctions you have of liver or muscle, that's pretty specialized. You can't redo that very often. However, there's a lot of discoveries. You had this population, roughly a third men, you had a third postmenopausal women, and a third premenopausal women. And so the simple summary was kind of surprising in many ways, that most men, given what they call the top level, the level of choline that was recommended by NIH, which is 550 milligrams per 70 kilogram body weight, which is kind of what I am, by the way. So at that amount, most men didn't get enough choline. 6 actually had dysfunction at the repleted level, at the level that everybody's supposed to be healthy at. So then when they went down to a far lower level, they all had problems. And so they would measure it in terms of, did you have liver problems? Did you have muscle problems? Did you have liver and muscle problems? And that was the kind of the category. What they found was at the repleted level, postmenopausal women were fine, premenopausal women were fine, but when they dropped it down, they found that the postmenopausal women suffered and the premenopausal women did fine, except for just a few. And this study was so profound actually. This study opened up the door to saying, so what is the difference between these people? These questions aren't asked much. So for the past 70 years, if not 100 years, the idea that, oh, well, yeah, you know, in this group, some do better, some don't do better, and just left it at that. This was, well, some did worse, some did okay. What's the difference? So now they found that they had certain mutations. And as a stereotype, which is never the case, premenopausal women have the ability to generate their own choline. So it really doesn't matter that they eat choline. It does, but they have this buffer back there. Those who have a genetic problem, mutation, SNP, can't do that. So they're going to be suffering through this high estrogen time as if, and not be able to generate their own choline. So they're 100% dependent on getting choline for themselves, for their own brain and nerves, but also should they conceive and have a child for that forming baby. Because if choline is deficient especially through the first month of pregnancy, it's associated with things called neurotube defect, which is open up the door. It's a number of different things, but we just call it spinal cord problems, brain problems of various types, and generally doesn't work out very well. And so this is critical. So if the mother can't make it and she's not eating it, the priority is going to be that the growing fetus, the growing baby, is the high priority. It's going to be extracted from the mom and given to this growing fetus. And hopefully that will be enough, that there's some reserve in the mother. And that's usually a process for a lot of different things, like calcium and magnesium, other minerals. The mother is mined she's the reservoir for the growing fetus, the baby. So that's really interesting. Next, though, when we talk about choline, choline does three things. It helps with this thing called methylation, which is all about the Judi experiment and the Agouti mice and all that change. It was turning genes on and off, right? So in the Judi experiment, it turned all these genes on that should have been on, and the other ones were off, and made healthy next generation mice. Okay, so that's one function. The other is it's responsible for making phosphatidylcholine in the body. So 95% of choline in the body is, in terms of phosphatidylcholine, lecithin. And that's the reserve. That's sort of the backup, all right? And so that's all the fat that surrounds all the cells of your body have lecithin in it. Big deal. It's very important. The third thing is, it is a precursor to making DHEA, one of the two most important omega-3 fatty acids. And DHA, as an omega-3, is 20% of your brain. All right, well, that's interesting. So if you don't have choline, you're going to be behind on making DHEA. You can say, well, I'll take fish oil. That's not a bad backup, and that's a good thing to do anyway. But what happens is, when you think about dementia and Alzheimer's, one of the obvious things that you see pictures of all the time is that part of the brain just disappeared. Part of the brain looks like it was eaten away. It wasn't so much eaten away, as it stopped having the things that allowed it to produce the fats that make up the brain. Interesting. So that's the third virtue of choline, that it makes DHEA for our brain. It's brain fat, if you will. It's cell fat, and it's for methylation. So when you think of choline and all its deficiencies, and that I think it's 94% of the United States is, by NIH's standards, deficient in choline, it's pretty important to consider the sources that you need, which come down to egg yolk and liver and their animal products, because it's more about animal cells than it is about plant cells. But it's pretty profound. And so when you add in the layers, the two layers of the methylation, the Judi's experiment, that he gave the methylation factors, basically folate, B12, choline, SAMe, which is basically a choline derivative, and maybe even beta-ene, which is still a choline derivative, and he changed the whole subsequent generation. That's like us. We are the Agouti mouse right now, and we need to take our methylation factors, three primarily. It's the B12 and the folate and the choline, and we need to get them through food first. Yeah, you can take supplements, but primarily, if you don't take food first, you're going to be missing a lot of other cofactors like B2, all the other Bs, right? And essential fatty acids that we've talked about. So it's pretty important We're the Agouti mouse, and we're like looking into the next generation, as well as how are we getting older? So I'm 66, I got another 30 plus years to live. I have to think about the fat in my brain, the neurotransmitters in my brain, neurotransmitters in my muscle, in my liver health, without being too obsessive about it. But these are big stones, big topics to think about and to address. Until next time. So if this is something that you're interested in, that is a topic that I obviously go deeper in terms of labs, in terms of how to do it, in terms of why you would want to do it, various topics, as you've seen that I've done in the past, then please let me know below in a comment. Till then.